Welcome to week number 3 for plant biology for fall of 2019. Hope everybody had a good Labor Day weekend. We had good weather here in Iowa, so successful all around. This week our reading is going to be in Concepts of Biology, Chapter 3. Chapter 3 is really talking about cells and how we understand how cells are made up, and how then that develops into the broader development of multicellular life, of which many plants are. We can really start separating out cells into two main different types. We can talk about organisms that are single-celled, so they only have one cell that makes up their entire organism, or we can talk about organisms that are multicellular, or organisms that have more than one cell, and that could be two or many. You and I are both multicellular organisms. So understanding the difference between single-celled and multicelled is important. Also understanding how we look at cells is important. A major component to scientific inquiry is understanding how these small particles of life function, and we use different tools to understand how they work. And we have three sort of main types. We have what are called compound light microscopes, which are sort of what you usually think about when you think about a microscope, and that's for looking at things like this. So glass slides, usually they're prepared by a company, but certainly if you're going to go into the medical field or something like that, you'll learn how to build your own slides and things like that. If this was a face-to-face course, we would definitely be building our own slides and looking at those slides through the microscope in lab. We also have what are called stereoscopes. They're basically, they work pretty much like a set of binoculars that are sort of fixed from the surface, and they're really for looking at larger structures. So if we have a large piece of wood, for example, and we want to see what the structure on the inside here sort of looks like, not at the microscopic scale like these, but at something maybe just below what we can see or what we can't really quite make out with the naked eye, we use what's called a stereoscope. The third main type of microscope that we have are called electron microscopes. Those are for looking at very, very tiny things. Electron microscopes can find some really great images, say of pollen, and our understanding of how organisms are related to one another can look at the pollen structure and see how those shapes change, and just utilizing a different tool for looking at a different scale of understanding. Through the study of cells, science has really developed what's called the cell theory. And the cell theory is one of these sort of foundational concepts in biology and the biological sciences. And that's really that all cells are derived from previous cells. So the cells that made up this tree grew from cells from the tree in the past. And so understanding the cell theory and how all of those cells are interrelated is a foundational theory for biology. The linkages that theory can build on is pretty interesting. Looking at, say, the evolutionary history of organisms, just the developmental aspects of individual organisms. So there's a wide range of things you can look at with cells. Point 3.2 of Chapter 3 is really sort of setting out the differences between prokaryotic and eukaryotic cells. In this class, we're going to be more focused on the eukaryotic cells. These are cells that are more advanced. Eukaryotic cells can definitely be single-celled organisms, can be eukaryotic, but they have a certain set of criteria that they have to meet. And so understanding the similarities and the differences between prokaryotic and eukaryotic cells is important. When we move into looking at eukaryotic cells, it's important to understand the basic structure of a cell. Plant cells have a slightly different structure than do animal or other cells. Again, understanding the differences between these is important, so being able to compare and contrast animal cells and plant cells is important, so that when we're talking about plant cells further on in the semester, when I say things like the cell wall, you have a solid understanding of really what sort of structure a cell wall is and how that relates to the rest of the cell, and then other organelles. That's a term you should know. Other organelles, how they relate to one another as well. So understanding the basic structure of eukaryotic cells, the cell membrane is an important structure and component that you're going to need to spend some time on, and it's a fairly it can be a fairly sort of nebulous concept because the way that our understanding of what the cell membrane, how it's structured, is it can be a strange sort of concept. So spend some time with that, and if you have questions about how the cell membrane is structured and what it actually means for cells, please post in the discussion, and I'm happy to talk until you're ready to throw up about the cell membrane. And also to spend some time and look at the major organelles or small components to an individual cell. Some of the organelles are specific to plant cells. Focus on those. Especially the major sort of metabolic ones and the really big sorts of structures inside the cell. Moving into 3.4, 3.4 talks about the cell membrane in specific. So they're really going to start talking in some depth and detail about that. Again, I mentioned that that's going to be a fairly kind of strange concept to work around. We can work through that in the discussion, and there's a lot of really good animation and images and things like that available online, and a simple Google search will find some really high quality images for that, as well as what is in your textbook as well. 3.5 is talking about movement of things in cells, primarily looking at passive transport mechanisms. So when we're talking about diffusion, osmosis, and a concept called tonicity, those are really big concepts that you're going to have to understand. Understand how things move from one part of a cell or between two different cells to another. Those would be what we call things moving with a concentration gradient with diffusion. If things are moving through a cell membrane, or a selectively permeable membrane, which a cell membrane is, that's a concept that we call osmosis. Tonicity is important because it's talking about how much water and salts, these sorts of water and salt balances, are keeping the amount of volume in a cell at a regular sort of amount, so that you don't have these cells explode or shrink down to the point where they no longer function and die. This is an important concept with plants. If you think of green leafy plants, when they start to wilt, the tonicity of those cells in that plant is decreasing. So understanding how water plays a role in the physical structure of plants and also in moving between different parts of the plant is important concepts as well. 3.6 is looking at active transport, which is moving things against a concentration gradient, and those are really important sort of biochemical and physiological concepts that you're going to have to understand. Again, as you have questions about these things, drop these questions into the discussion forum, and I can address them one-on-one, or you guys can share your own insights on these things. When we're talking about cells, again, we're going to be looking at things that we can't really see with the naked eye, and so understanding that the scale of our understanding here is different than you and I having a conversation. So chapter 3 is going to sort of wrap up our sort of general biology part of this semester. And after the first 3 weeks, we should have a pretty good sort of common language on which we can build and start talking about more plant-specific things, since this is indeed plant biology. I really like talking about cells. They are fundamental to our understanding of biology, and I hope you enjoy Chapter 3. If you have any questions, please post in the discussion forum, and we've already had some pretty good discussions in there already. So look for Chapter 3's assignments and discussions, and have a great week.